Good afternoon, all of you. I welcome.
You are not audible, Ritesh.
Hello.
Yeah.
Yeah, good afternoon. I welcome all of you in this sixth lecture of this online webinar series, Jigyasa. Today we have with us Dr. C.K. Katiyar sir to discuss on the topic the pharmacopoeial parameters of standardization of Ayurvedic methods from the industry perspective. Before starting this lecture, I Anupam sir for welcome and for Sri Ganesh of today's lecture. Anupam sir.
Thank you, Dr. Karunanidhi. And on behalf of entire department of Rasashastra, Kalpana and I welcome to all of experts, teachers, my dear students, and whosoever has joined this platform. And I definitely, of course, I welcome on behalf of my entire team to today's speaker, today's expert, Dr. C.K. Katiyar, sir. And I'm thankful, sir. I'm thankful to you. It is my proud privilege again to welcome you, sir. Before we start this webinar, just want to share that I had been guided, I had been supported by today's experts many years before, more than 14 years, if I'm correct, sir, correct me. Since 2007 and 6, he had been always guiding, supporting, mentoring. When what any a time he is always available to support us, and I'm fortunate enough I also got the same discipline and the same institution from where the sir has taken the education from the BHU. And before to talk anything about the pharmacopoeial parameters, standardization of the Ayurveda products, and all details, it will be not relevant for me because we are here to to the expert, to Katiyar sir. I will not waste any more time, and I will request again to Karunanidhi to please brief. Everybody knows, sir. He doesn't require any introduction, but as a customary, but as per our defined views, we again request you, Dr. Karunanidhi, please briefly explain about the bad sketch of today's speaker. Then the sir could start his derivation. I will not come in between you and sir and the audience. Over to you, Dr. Karunanidhi.
Thank you so much, sir. Uh, Before starting the lecture, I would like to just briefly introduce all of you to the sir, although sir is very known personality among all of us and Ayurveda community people. As Anupam sir also told, sir has completed his MD and PhD from IMS BHU, and the very most important part, most attractive part of sir's CV, I could say, sir has worked more than 30 years in this research driven companies like as directors, served the directorship in the Ranbaxy Laboratories Limited, Gurgaon. Sir has head the Herbal Drug Research Division of Dabur India Limited. Sir was the Vice President of Dabur Research and Development Center, that is in NCR, Delhi. Currently, sir is working as CEO, Healthcare Department of Pharmacy Limited in Kolkata. Sir has also contributed in the quality control part of the Ayurvedic industry, being the member of Ayurvedic Pharmacopoeia Committee, Herbal Committee of Indian Pharmacopoeia. Sir is also Southeast Asia expert on the dietary supplements for US Pharmacopoeia. Uh, Sir is also a member of editorial board of the reputed journal like Journal of Ethnopharmacology, Frontier Journal of Pharmacology, and Journal of Ayurveda and Integrated Medicine. And his Also the member of ASU Technical Advisory Board, governing body of All India Institute of Ayurveda, and the Institute of Bioresources and Sustainable Development under the Department of Biotechnology. Sir's work is also recognized by several awards and recognitions. Sir is awarded by the membership of National Academy of Sciences India and Fellowship of Indian Pharmacological Society. So, sir, we welcome you. Uh, for any drug, first thing the drug should be safe, second the drug should be effective, and if it is having both thing, it should be reproducible. So, to reproduce any drug, we should have to have universal standard. Today, discuss on the pharmacopoeial parameter crystallization of Ayurvedic product. We'll listen to sir. Sir, welcome you. Over to you, sir. Thank you.
Thank you, Dr. Anupam. Let me first upload my presentation. Is it visible?
Yes, sir.
दिख रहा है?
जी.
एक मिनट. अभी दिख रहा है?
Yes, sir.
Okay. So thank you, Dr. Anupam, and for giving me an opportunity to be on this forum. I think probably for the first time. And in today's perspective, when Dr. Anupam asked me to speak on a topic, the most dear topic to me is the standardization parameters, following pharmacopoeial parameters, and what we have learned during my 30 years, where we are and where we need to go. So today I will not be discussing only about the pharmacopoeial parameters, but I will try to go beyond that. So, but before going to pharmacopoeia, all of you must be aware that today the turnover of Ayush is, as per the Ministry of Ayush data, is close to $18 billion. And the kind of products we are dealing with, extracts, phytoconstituents on one side, to pharmaceuticals, to veterinary medicines, to proprietary products, to cosmetics, FMCG, classical Ayurvedic products, herbal extracts, nutraceuticals, all kinds of total spectrum. And the total, and it is also, but the $18 billion also includes services. So it is $18 billion is including services and products both. If we look at the exports, so if China controls almost 30%, India is way behind. India is controlling only 11% of exports of medicinal and aromatic plants. But if you look at the total growth of the sector, as per the report of FITM, it was Ayurvedic sector which has grown highest with a 12% in the last 5 years, from 2012 to 2018. So the growth is there. Therefore, the trust of people on Ayurveda and Ayurvedic products has grown tremendously. Now I will come to the today's subject of pharmacopoeial parameters. But before going to pharmacopoeial parameters, let us see what is pharmacopoeia. A pharmacopoeia is a compendium laying standards are medicinal and pharmaceutical substances and formulated preparations. It is the recognized authority issuing the book of standards is appointed by government of a country. Every country is supposed to have its national pharmacopoeia. Objective of the pharmacopoeia are to control the quality of the product in terms of three things: identity, purity, and strength, and therefore to ensure quality, safety, and efficacy of the product. If you look at Ayurvedic medicines, the resources it uses 90% to 95% are plants, 2 to 3% of minerals, 2 to 3% of metals. This is if you look at the in terms of volume. And animal byproducts and marine products are very small, 1 to 2 products. This is old slide. Till that time, only 8 volumes are published. So there were 600 monographs in the 8 volumes of products, where 8 volumes are dedicated to water and hydroalcoholic extract. Later on, volume 9 is also dedicated to that only. Now, if we that was a single herb monographs. If you look at the compound or formulations, this is also in old data. Up to volume 6, it had covered 152 Ayurvedic formulations, which are basically taken from Ayurvedic formulary of India. So their quality standards of all the dosage forms are like Savarishta, Ableha, Churna, Bati Gutka, Guglu, all of them are covered, but there is long way to go. We have hardly covered not even 20% of the formulation so far. Coming to today's discussions, I have certain points to ponder. So today it will be more like a loud thinking. So what are the pharmacopoeial parameters of quality for products and single herbs? Where do we stand today? Then everybody is talking about the phytochemical marker compounds for standardization. We will look at their current status. Nobody in the pharmacopoeia is talking about the negative marker compounds. I will share one specific example on that also. While we are talking about the marker compounds, most of them are analytical marker compounds. But can we look at the biologically active marker compounds? And if yes, how to identify those compounds which are biologically active? For this, are there some quicker methods for efficacy evaluation? We'll look at that. We also need to have awareness about the herb-drug interactions. Going beyond pharmacopoeia, we are very slow in Ayurvedic industry, very slow in technology adoption. Probably one of the reasons is the current rules and regulations also do not permit too much adoption of technology. And then the lack of adoption of latest pharmaceutical dosage forms, because the pharmacopoeia does not permit you. Pharmacopoeia limits only to those dosage forms which are mentioned there. But if you go beyond pharmacopoeia and proprietary, there is a lot of scope. But there again, there is lack of adoption of technology and some other issues. So let us first talk of the products and pharmacopoeia. So if you see on the left side, they are the total pharmacopoeial parameters for any churna. But they are only the product. If you look at the right side, you see it is our proprietary product Jandu Nityam Churna. We are conducting 234 tests on every batch. Out of it, 135 are on the raw material, because that raw material depends how many raw material you are using, multiplied by 10 or 15. So if you look at the bottom here, heavy metals, aflatoxins, pesticide residues and microbial load, they are tested on every batch, including raw material. So 135 tests on the raw material, 67 tests on the packaging material. In process control and finished product contains 234 total tests, some of which are pharmacopoeial, some of which go beyond pharmacopoeial. But when we look at the raw material, most of the tests are pharmacopoeial. This number becomes high because the number of ingredients go up, number of tests also go up proportionately. So if you look at the, for example, Sudarshan Ghanvati, which has some 53 odd ingredients, the number of raw material tests go up to 621. So total number of tests a laboratory has to do, a quality lab has to do on product like Sudarshan Ghanvati, the number is 729 tests. If you include packaging material also, even if you exclude packaging material, even then the number is almost 650. Quite big number. And look on the left side, they are all the parameters. Some of them are from pharmacopoeia, some of them are in addition to the pharmacopoeia. This is about the finished product. Now, if you look at the single herb monograph, there are the descriptions First, we need to describe the macroscopic powder description. Then there will be TLC. Then there will be some quantitative parameters, which are foreign matter, loss on drying, total and insoluble ash. Then there will be some, in certain cases, the assay of marker compounds. Then there are additional requirements like heavy metal, microbial contaminant, pesticide, and aflatoxin. We will look at some of the relevance of the marker compound. I wish to discuss some of the data with you which people normally shy away, but this was the published data that out of 3,486 number of herbal raw material tested for pharmacopoeial standards, almost 11.5% samples failed in quality parameter. So 401 out of 3,000, almost 3,500 samples failed as per a data generated by PLIM, the Ministry of Ayush. Now, we also have done at our company at Jhandu, we have tested 2,607 samples. So it is better we look at here. We have tested 2,637 samples in the last 3 years, which cover 134 species, out of which 228 samples were rejected, which is close to 9%. And they are the how many roots, how many whole plants, how many fruits, extent that the breakup is given. But the matter is that almost 9% samples were rejected. This is pharmacopoeial quality parameters for API. If we go deeper into it, what were the parameters on which these samples failed? If you look macro and micro description, highest number. And if you look at data for 3 years, 2018-19, 19-20, 2021. This data normally you will not find anywhere, but I asked my team to publish it, and so it was published in 2021, this data. And in foreign matter also a lot of products fail, but in foreign matter what is included? If you look at the definition, if you look say for example tulsi leaf, so leaf के ऊपर एक छोटा सा जो में डंठल होता है, even that is considered to be the foreign matter, and its limit is 2%. Should not be more than 2%. So a lot of time, because tulsi was used during pandemic in huge quantity, so these kind of foreign matters are sometimes going and the matter fails. But then what happens is you do the sorting. So instead of rejecting whole batch, you do the sorting and then use the right material. Similarly, if you see loss on drying, so moisture content, then it also failed was water soluble extractive and alcohol soluble extractive, few samples. So these are the various, and all of them are pharmacopoeial parameters, which are description, foreign matter, loss on drying, water soluble and alcohol soluble extractive value, total ash and acid insoluble ash, and in certain cases volatile oil content also. So if you see here also, we have seen that in particular case year-wise, if you see, the failure rate was something around 11 to 12%, which is matching same with the PLIM data. Now these rejections are of two types. One is remedial parameters on which the product fails. For example, description, foreign matter, loss of drying and insoluble ash. If it is failing insoluble ash, you can just do some dusting and dust goes off and it matches the quality. Loss on drying, you just dry it a bit more, it will match the requirement. Foreign matter, you do some kind of cleaning and that also can be taken care of. But there are certain non-remedial. So these remedial parameters, the product can be used and reused, can be corrected and used. But if it fails on water soluble extractive or alcohol soluble extractive or assay content, the only way is to reject this material. So these pharmacopoeial parameters fall under two types. One is remedial, other is non-remedial. For non-remedial, the only way is to reject the material. For remedial ones, you can reuse the material. And some of these can be taken care of if we follow good agricultural practices, good collection practices, and good post-harvest practices. It can be managed. So this is the same data what I was just sharing. Now, when we looked at the heavy metals, we found that in case of heavy metals, almost 10% means when we tested these compounds, almost 20 out of 183 samples failed. But to my surprise, 10 samples failed only on cadmium. It was some new finding because before that, in my experience from Ranbaxy and Dabur also, no sample ever failed in cadmium. But now suddenly samples have started failing in cadmium, which is a red herring. And I need to go deeper into it. I think it was some plant of the not Brahat, some punch, some the small plant it was, which was responsible. Mostly only one plant. So other if you remove that, the rejection is close to 3%. Otherwise, it is 10% on heavy metals. You might have remembered last 2 years, about 2 years ago or 1.5 years ago, a paper appeared on report of hepatitis by use of guduchi. And certain doctors said that they had to transplant the liver. Ministry of Finance, all of us also reacted to that. The data may be wrong. They might have not used the right quality of the herb, right species. Ministry of Finance also came out with the advisory that in case of Guduchi, use only Tinospora cardifolia because the adulterant, the cases which have reported to cause liver damage might actually have used Tinospora crispa, species which is found mostly in South India. And they also came out with the advisory that no company should use crispa, and they provided some morphological characters. But the problem is when you have got an extract, suppose some company using the Gluchi extract, there is no morphology, there is nothing to see a morphology about. So what was required is there some very specific marker compound which is present in crispa but not found in cardifolia. So we started working. We said that since we are getting a lot of extract from outside instead of using crude gluchi, so our problem is how to identify. And there was no guidance available from anywhere. So what happened was we checked a lot of literature. We found that Professor Ikhlas Khan from Mississippi had worked on the 10 species of Tinospora cardifolia, and he reported that there is a compound called borapitoside. Borapitoside is a compound which is specific to Tinospora crispa and is not found in Tinospora cardifolia. Second issue was where to find this compound? Nowhere. So I asked my team of phytochemists. They spent 6 months time, and only last month they were able to isolate this compound borapitoside, and now you are looking at this TLC plate where if you see T1 is crispa, T2 is borapitoside, the compound which is specific to crispa, but if you see on the T4, T4 is a Tinospora cardifolia. This compound is not available here. To be more specific, look at the last picture where it is the light blue color is borapitoside. It is seen in the Tinospora crispa, but you cannot see the blue color compound in Tinospora cardifolia. So now from this month we have started using borapitoside as a negative compound, while cardiofolioside is used as a positive compound for this Guruji. And probably we are the only company in India which have been able to isolate, and we started using it in-house for correct standardization and using the negative marker compound. Now, let us come to the very, very crucial point. Since most of you are students of Dravyaguna also and besides Rasashastra, let me share with you. So, when Ayurvedic Pharmacopoeia, if you remember, up to volume 6, there was no chemical, phytochemical compound was mentioned up to volume 6 because all the parameters were physicochemical. But after that, when Ayurvedic Pharmacopoeia committee started looking at the marker compounds, then some very reputed phytochemists were involved in the committee and they started giving suggestions. So what happened was when they looked at the marker compounds for these plants, what is ideally do? Everybody does that only. So you look around you, what is the literature published on the chemistry of those plants? So you look at the compendia published by CSIR or the various journals of phytochemistry and start looking at the compounds which were isolated from the part of the plant at the most. And we start recommending the marker compounds for those plants. But at that time, we forgot one thing, that whatever compounds were reported and whoever had reported, they did not isolate those compounds to standardize Ayurvedic medicines. Those compounds were isolated for somebody doing PhD in chemistry, somebody doing PhD in organic chemistry or natural porous chemistry, to isolate a hitherto unknown or new chemical compound from a plant so that they can get a good publication. So chances of getting such compounds are more with either methanolic extract or hexane extract. So what happened? Most of those compounds were isolated from methanolic or hexane extract. None of those compounds were isolated from water extract. While the fact is, in Ayurvedic medicine, we are using mostly water as solvent. Now, what is the impact? Now let us see. So what we have done, we have looked at the Ayurvedic Pharmacopoeia of India, how many marker compounds have been mentioned. So we also have done some exercise. The same marker compound has been mentioned in the crude raw material, in the water extract, as well as in the hydroalcoholic extract. But when we looked at their solubility, we found only 4 compounds, out of 30, only 4 out of 30 compounds in Ayurvedic Pharmacopoeia of India were water soluble. So therefore, the chances are that if you are trying to find out Gugulu, Sterone, E and Z in Mahayogra Gugulu, you will not find it. The reason? Because E and Z Gugulesterones are soluble in acid state, they are not available in water extract. So a drug inspector will fail your sample of Mahayogra Gugulu saying it does not contain the marker compound mentioned in the pharmacopoeia, therefore your product is substandard. My God, the marker compound selected is not even water soluble, and we are using same marker compound has been recommended for in the crude raw material. How is it possible? Okay, move forward. We also looked at the Indian pharmacopoeia. In Indian pharmacopoeia, so same thing, only 4 out of 44 compounds were found to be water soluble. So for example, if you look at Haridra Khand and try to test it for curcumin, do you think you are going to find curcumin there? Very small quantity, negligible maybe. So therefore, and when we did that, I said, okay, let's do one thing. It is just theory so far. Let's do some practical experiences. So what we did was, we tested our Jhandu Pancharishta which contains some 35 plants. And we used 32 marker compounds, you will not believe it. We tested these 32, 21 and 34. So we used these 34 marker compounds, and we also noted down their presence in the crude raw material and their presence in the finished product. We could not find even 50% of these marker compounds in Jandu Pancharishta, despite the fact they were mentioned in the pharmacopoeia. Okay, leave Jandu Pancharishta, which was hydroalcoholic formation. Can we do the same thing in Chyawanprash? Also, look at it, same thing here. So Chyawanprash also contains almost 38 herbs. We conducted same kind of exercise here also, and we tested 17 and 30. We tested 30 chemical compounds, 30 marker compounds we tested as per the pharmacopoeia, and we could find, I think, almost 13 or 14 only. So therefore, what I am saying, that the selection of marker compounds in Ayurvedic pharmacopoeia of India needs to be reviewed, and the whole chemistry needs to be repeated using water extract. So the gap analysis is therefore based upon the objective respective PSCs. Effort was to obtain compound. Most of the compounds were not detected, and therefore what we say now, that whole chemistry needs to be repeated from water extract. And you see, there was a detector called ELSD. So most of the compound, normally when you buy SPLC, you buy UV detectors, because most of the compounds which come from alcohol, they are sensitive to the UV. But water soluble compounds most of the time are not sensitive to UV, they are sensitive to other things. So the another detector called ELSD. So therefore, when I say that we should use water extract as the starting material and repeat whole exercise, therefore Ministry of Ayush should consider setting up a dedicated institute, Ayush Institute of Chemistry and Biology, dedicated to this kind of thing, where water extract shall be used for total chemistry, and what to be done by biology, I will come a bit later. So that was about the chemistry and the marker compounds. Since I am talking pharmacopoeia, so let us go deeper into the pharmacopoeia. Now let us look at the biology. So these marker compounds, whatever have been mentioned, hardly 20% of them are reported to have some kind of biological activity. Hardly 20% of them. Most of them are not. So there is some short method what we call bioassay. So what is the need here is that we don't need animals here. We can evaluate the efficacy in very short time, and not only extract, we can also test the efficacy of the pure compounds, which later on can be so pure commonly known marker compounds can also be subjected to this test. And if they are bioactive, we can know bioactive marker compounds also, and they are also used in the high throughput scanning. So how are they identified? These bioassays, how are they developed? Look at the literature and look at the product solubility profile is required, and then what are the systems required. All this is published, is available in the literature, and then we need to conduct some performance tested materials which are positive compounds, positive or active compound, and then test compound, and then determination of IC50, EC50 or CC50 values, and then look at the potency. So these bioassays, look, if you remember, Dr. Mashelkar long ago said that for modern medicine, our approach has to be from mouse to man. But in case of traditional medicines, this approach has to be man to mouse. So efficacy is man established. You need to conduct mechanism of action, other studies, then go to mouse. So therefore, what we are saying here is that these bioassays can be used in case of classical medicine for validation of efficacy of the product, but in case of proprietary medicines, they can be used for substantiation of the claims. These are some of the bioassays what we at our Imami and Jhandu R&D lab are having. Antioxidant, we are having ABTS and FRAP. Anti-aging, we are having advanced glycated end products estimation. Herb drug interaction studies also we are doing, and probably we are the only company in India which most of the new proprietary medicines are subjected to herb drug interaction before launch in the market. We have certain assays for in vitro and enzymatic assay. So for diabetes, for skin health, we have PD5 alpha inhibitor for as aphrodisiac. For memory enhancement, we look at the acetylcholinesterase. So just to explain, say in our brain, it is the acetylcholine chemical which is responsible for memory. In the old age, the production of acetylcholine goes down. So only two ways. Either you increase the production or you decrease the destruction. So there is an enzyme called acetylcholine esterase. So if you reduce the activity, inhibit the activity of this enzyme, then acetylcholine will automatically go up in the brain and it will reflect in the better memory. So we are conducting acetylcholinesterase inhibition assay for all the Medhya Rasana products. Anti-inflammatory, we have LOX and COX assays, and these are enzymatic. We also have a cell-based assays. One of the most famous is natural killer cell activity, what we are having. For diabetes, we have glucose reuptake assay. For allergy, we have. For obesity, is not very effective, but some assay we are having. And we also have generated some methods for wound healing. And for bone health, we have osteoblast proliferation assay using cell lines. These are our in-house assays. Now, if we have to do some assays, so these are the ABTS and FRAP, LOX and COX, anti-diabetes, NK cells. Giving some examples, so we have a product called Diabrista. It's a Arista for diabetes, and it has shown very good activity on rat L6 myotubes as well as alpha amylase and alpha glucosidase inhibition activity at 8.73 and 10.93 IC50 value, which is supposed to be very good. It has been published also. When you look at the Chyawanprash, we have got data on the FRAP antioxidant assay 24.9, ABTS 12.9, and best anti-aging, anti-glycation assay 4.7, very potent. NK selectivity 3 times more immunity power. Similarly, it's also we have data on memory, so acetylcholinesterase inhibition and allergy. So we have histamine release also. So we have conducted all these assays on Jandu Chyawanprash. This is with Jaggery. So this kind of thing we keep doing at our laboratory. But we look at the classical products like Mahagra Joglu. We have done the 5 LOX assay, and 8.32 is a very potent, because anything less than 20 microgram is supposed to be potent in Ayurvedic medicine, because mostly we are using extract of crude herbs. Now, after this, our pharmacopoeia discussion is over. Now, if we want to think a bit more going beyond that, so our title said where we are and where we need to go. We have just shared where we are, but where we need to go. So we need to identify relevant analytical and biologically active markers. And this is now feasible because of the new technology available. Like I say, bioassay, which was not available 10 years ago, but now they are available. So all the marker compounds can be subjected to these bioassays, and currently known analytical markers, some of them can certainly become the biologically active markers in future. Second is we need to give a total relook at the marker compounds, isolation methods. The current marker compounds, as I told you, 90% of them are not applicable when you test them in the product. Biggest worry is that if a drug inspector starts testing your product for the marker compounds in pharmacopoeia, he will fail the best quality product also without because of no fault of the companies. Next is introduction of herb drug interaction should be there in the safety protocol. So because now the technique is simple, it should be part of the safety studies now. And you are the students of Rasashastra and Dravyaguna. You know the churna is the simplest dosage form, but to me it is the most complicated. Trifla and ashwagandha. Trifla and ashwagandha churna are more susceptible for microbial load. You pick up any product from market or your own pharmacy and you test for microbial load after 6 months. You will find the limit is 1 lakh, you will find 5 to 6 lakhs. Only two companies in India, Dabur and Dhandu, their products you will not find, they always pass. The reason is these two companies are subjecting all their powders to gamma radiation, which is permitted by Ministry of Ayush for decontamination. So there is a need to develop and identify inexpensive, effective method of microbial decontamination. UV exposure does not work. The only method which works is the steam distillation method, steam steam exposure method. But there at the end of the day, the moisture of the powder goes up and it will remain sterile for a few months. But then again, there is chance of higher microbial load. Right now, gamma is the best method here. Some of the things which industry requires is development of natural foaming agent. So you are using shampoo, you are using toothpaste. Both of them use foam. So SL is the chemical compound. We need to do lot of research, and I am sure that in Ayurveda they are having lot of leads for this. For like Rita is one, and any plant containing saponins will be even Brahmi can, for that matter, can become a source of foam. So development of natural foaming agents, and most important, development of natural preservatives is the need of the day. And as you know, right now, lot of people are using lot of flavors and perfumes. So nature identical flavors and perfume development. These are the some of the areas which are directly not related to Ayurveda, but at the end of the day, they are going to be used in Ayurvedic products, and therefore they are important. So the scope of research in Ayurvedic products need to expand beyond what we are doing. From active, we also need to expand to the inactive or what we call excipient areas. Right. So that was today. So in conclusion, a lot has been achieved regarding raw material and finished product standardization for herbal products. But one more thing about the metal-based product. So these almost no work has been done. Almost no work has been done. Very rudimentary stage. So lot of work needs to be done in Rasashastra, especially Bhasmas and Rasashadhis. A high level of interdisciplinary work is required to make this. Approach of product standardization needs to be more relevant and inclusive. And when we say inclusive of both Ayurvedic method of processing and the area of chemistry and biology has to be in consonance with the Ayurvedic method. These two things cannot become 36. They had to become 63. Look towards each other, not looking away from each other. Right. So with this, I am very thankful to Dr. Anupam for giving me an opportunity to be with you today. And now I will be looking forward to any questions and answers. I am sure a lot of students, researchers might be having some questions. So I am happy to reply that. Thank you very much.
Thank you so much, sir. It was very interesting lecture. मजा आ गया. Thank you for throwing the lights on the importance of pharmacopoeial parameters, including remedial and non-remedial parameter. You also highlighted about the analytical, biological, and most important, the negative biomarker compounds, then in vitro bioassay, and at the last you also mentioned about the need of the what is the way ahead of the industry. Thank you so much, sir. We do have some question, and the house is also open for the question and discussion. If anyone into who is joined the meeting have any question or query, they can put now, maybe in the chat box, or they can unmute yourself and can put the question. After that, I do have some question from myself and my friends. I'll ask that. Anyone from the meeting? Okay, sir, I'll ask then if there will be a question from anyone, they can come in between. Sir, a very simple question. They say for in-process quality control, you also mention about 2 or 3 of product of Jandus you are doing a number of in-process testing. But for a simple formulation quartz, हम क्वाथ बना रहे हैं सर. क्वाथ के लिए what could be the appropriate in process quality testing? क्योंकि people do bricks. ब्रिक्स तो अगर वो ऊपर नीचे भी है उसका, तो ब्रिक्स में बहुत चेंज आता नहीं क्वाथ में. दैट बट आई हैव सीन इन प्रैक्टिकली. सो व्हाट कुड बी द appropriate in process testing for quartz?
So in quartz you need to ultimately, तो आपको ब्रिक्स पर ही रही करना पड़ेगा ultimately, because that is what you can measure. Suppose you are doing TLC, competitive TLC, and if there is some method of HPTLC, you can have some kind of quantification. But the problem with that is that every 5 minutes you will find changes there. Every 5 minutes. But if you do bricks, at least there is something which is constant. You can stop with the bricks. So I think bricks is the at this point of time the most appropriate. But once that quartz becomes converted into a finished product, then you can apply other products like I say of the compound, and so many things can be done.
या राइट सर. क्वाथ का एज एफजी तो हम बहुत सारी चीजें करते हैं, इवन टोटल सॉलिड भी कर लेंगे तो विल बी हैविंग सम डेटा, बट आईपी क्वाथ का करने में थोड़ा सा इशू रहता है. एक और चीज सर, आपने मेंशन किया एक साइड वेयर रॉ मटेरियल वाज फेलिंग ऑन डिफरेंट पैरामीटर्स. एक सिंपल सा क्वेरी था, अगर मान लीजिए कोई हर्ब सैंपल है, इफ दैट इज फेलिंग ऑन मैक्रो डिस्क्रिप्शन, मैक्रो और माइक्रो, will it go for further process physicochemical testing and sophisticated analysis?
No, it will depend on two things. अगर तो आपको उसको यूज करना है, फर्स्ट ऑफ ऑल कि कितना रेंज है उसका फेल का. सेकेंड ये है कि अगर आपको उसको आगे भी ले जाना है, काढ़े में क्वाथ बनाना है, तो यू नो क्वाथ में जाएगा 100 डिग्री ऊपर जाएगा तो वैसे भी खत्म हो जाएगा.
राइट.
प्रॉब्लम तब है अगर आपको उसको बनाना है चूर्ण. फिर आपको चूर्ण को गामा करना ही पड़ेगा, नहीं तो वो पूरे प्रोडक्ट और को भी खराब कर देगा.
No sir, I am talking about description sir, कि मैक्रोस्कोपिक और माइक्रोस्कोपिक डिस्क्रिप्शन
ओके,
तो नेक्स्ट जाएंगे कि नहीं?
No, then you not go forward. अगर if it is falling at microscopic, then you cannot go forward.
ठीक है सर.
Because that is how you identify taxonomical identification. अगर आपका स्पीसीज गलत हो गया तो कैसे आगे जाएंगे फिर?
कैसे करेंगे? राइट सर. एक क्वेश्चन आया है सर, डू वी नीड टू चेक फॉर माइक्रोबियल लोड इन रसौषधि कंटेनिंग हर्ब्स एंड इंग्रेडिएंट्स?
No. Most of the rasashadis, because when you prepare, they are going up to 700-800 degrees, right? So there is no microbial contamination in that. And even in the pharmacopoeia, when I was member, we were preparing the pharmacopoeial standards for rasashadis. We deleted this requirement of microbial load. But the problem was that microbial load, pesticide, heavy metals, they are mentioned in the general notices. So anything in the general notices applies on all the products unless you write in that monograph that this test is not required.
Okay. But for the formulation like Arbamino, जैसे आरोग्यवर्धनी बटिया, for that तो we have to do that.
Yes, anything where you are adding herbal ingredients also, then you need to do that.
Sir, one more thing, जैसे we talked about, you mentioned about markers. So if we are doing a process like we are doing Snepak, okay, in that process maybe some of the active compounds they have changed to in the shape, उनका कुछ बदल गए हैं. So how we can map and quantify those who are changed into something else?
One PhD thesis, one or two PhD thesis has been conducted
Hmm.
at Jamia Handar
Hmm.
on my behest on Asavaristas, not on Snake Kalpana, but on
Hmm.
Asavaristas. That
Hmm.
we also are thinking that during the fermentation, some compounds are changing their chemical structure.
Hmm.
What is called chemical transformation taking place. So
Hmm.
there are papers published now that they
Hmm.
have identified two or three compounds, which were one was Asokarista, on which paper is published by Jamia Hamdard. Dr. Kanchan Kohli is the professor there.
Hmm.
So she had published that yes, there is some kind of chemical transformation taking place. And two most important thing, which most of us do not know, that all the Astavaristas contain vitamin B complex. Because vitamin B complex is mostly prepared, manufactured by fermentation process only. But the difference is all Astavaristas will have different composition of the different B complex. So it will not be same composition all. Ashoka Rista will have, so like for example, I have conducted a lot of work on that. Ashoka Rista is very rich in folic acid.
Interesting.
So therefore, so when I was, I think hardly 5 years in industry, I am talking very, I think 95 or so, we conducted a clinical trial on Ashoka Rista at KA Mumbai, and I was presenting data to some Vaidyas and some
Uh,
doctors. They said, the doc, we have noticed that Ashoka Rista, then I say, hemoglobin also goes up. How do you justify that? तो मेरे पास कोई जवाब नहीं था. So I said, because you are using lot of jaggery in that, and jaggery is also source of iron, so that may be leading to the this. But I was not satisfied. 15 years later, when I joined Ranbaxy, and when I looked at the actual fermentation process, then I
mm,
again tested Ashokarista. Now I knew that B complex is generated by fermentation. Then suddenly I realized that Asokarista is highest component of folic acid. So actually the hemoglobin going up with Asokarista because of folic acid, they're not because of iron. Right? So the very interesting, all the Asokaristas contain one or the other B
mm.
complex, which also need to be evaluated, need to be tested, and then the biological activity need to be defined around that. And Department of Bhaishajya Kalpana at NIA should take up the project on this.
Very
Uh,
interesting
uh,
information.
and you see some of the papers we have published. We published some papers on Draksharista,
Uh,
Ashoka. Some papers we have published from Dabar and Imami also. Not too many, but I think you guys can do much better job because we in the industry don't have too much time to basic research. But you can give the topic to some MD Ayurveda student in Bhaishajya Kalpana and look at the uh, complex composition and chemical transformation. Both of them can be done in the same thesis,
Definitely, sir.
right?
I- Definitely, sir. And those kind of research work, we would also we required your help and your support also.
I'm always available.
Thank you so much, sir. And one more question, sir. You also mentioned about the presence of cadmium. Even some of our samples were also failed due to presence of cadmium. Have you tracked or got some
Yeah,
points
yeah, yeah,
where
yeah,
the
yeah, yeah.
cadmium
No,
is
no, no.
coming
That's
nowadays?
I told you, sir, very surprising, very surprising.
Uh,
Because in the 30 years of my career, I saw some of the products failing in cadmium only last 5 6 years. And ये तो था ये शाल पर्णी, it was one of them. Either Shal Parni or Prasni Parni, one of them was failing. I don't remember now. That was only single plant which was failing. I think it was. I don't have full data right now, but it was one of them only. Either Salparni or Prasniparni.
mhm.
So maybe that is specific to it. Otherwise, if you delete that figure, then it's almost 3% failure only on heavy metals. And earlier also with the CCRS, when I was at Maxi, we conducted a study on almost 1,500 medicinal plants on heavy metals, because that was the time when Ministry of Ayush was supposed to set up the limit for heavy metals.
Mm.
I suggested that don't set up the limit like that. Let us first generate data. So we did generate data on almost 5,000 samples. 1,000 were done by Ranbaxy, 1,000 done by CCRA, some done by CSIR. And basis that only the current limit what you see, mercury we could not find data. So lead, cadmium and arsenic, that data was basis, that data what we generated in India. For mercury we could not have any resource. So we depended mostly from California, some data, so we kept only 1 ppm. But for other three, we had Indian data only. So the values are based by Indian research.
Yeah, we do also have seen the cadmium in some formulation basically, and we are
Right,
also
right.
trying out to track that from where it is coming. There
So
is one.
look at it might be Prasnaparni or Prasparni or Salparni. If it contains, maybe that can be one possible source of
We'll check.
cadmium.
Sir, one more question is coming into the chat regarding the using of natural foaming agent in shampoo or other cleansing formulation. What is your experience, and that also increase the cost of the product?
No, it is not. I don't have any product for that. I am saying it is the need of the country.
Yeah.
We should develop natural foaming agent. Otherwise, see, we have got lot of lead. We have Rita, we have Brahmi. If you buy lot of foam comes out. So some basic research is required. So right now, when we talk of development of Ayurvedic medicine, we mostly talk to develop the products, proprietary medicine. But no, we should now think of going beyond the medicine. Look at the development of excipients,
Right.
which is like preservative, like coloring agent, right? Like foaming agents, and so that's my suggestion. So right now we don't have any natural foaming agent, and anybody having natural foaming agent is the multi-billionaire of tomorrow. Just imagine how much shampoo, how much toothpaste.
Mm.
Everybody, nobody wants to use SLS as foaming agent, but
Yes,
they
right.
don't have any alternative.
Right, right, sir. As you mentioned about the natural excipient, I have also read some research paper where they have published the antimicrobial activity of these natural volatile oil, essential oils. What's your view?
No, you are right. See, in Europe now what happens is, for the veterinary, the
Mm,
use of antibiotics has been banned.
mm,
So all these essential oils
mm,
are now used as substitute of antibiotic
mm.
as antibacterial agents in veterinary practice in Europe.
Okay, sir.
So yes, they are good antibacterial agents, but we cannot use them as preservative. Because to become preservative, there are certain requirements. It has to be taste neutral. It has
Yeah,
to be particular solubility. It has to work at the minutest of the dose, right? So all these are failing on that account.
lot of work has to be done on it.
Lot of work has to be done.
I guess Anupam sir want to say something there. Hand is raised. Anupam sir,
Yep.
please.
Yes, sir. Namaskar, namaskar.
Sir, I guess you're on mute.
आपका म्यूट है आपका. नहीं अभी म्यूट है, अभी म्यूट है. सर अपना अनम्यूट कीजिए. सर
I guess
आवाज नहीं आ रही है.
not good, not very good.
आवाज नहीं आ रही है.
Let me just ask him to come back, sir. Anyone, any point in from the meeting? We'll move to what of things.
Karna, I am audible.
Yes, sir.
Yeah, now, but they are echo.
Thank you, sir. How are you, sir?
Fine, fine, fine. Thank you, thank you.
Namaskar, sir. Namaskar. Sir, actually, I have one question, sir. Uh, Basically, sir, regarding from the metals and minerals, actually. Sir, actually, we are facing one definitely problem. We have, sir, you have, sir, very large expertise, sir. Identification of the, sir, final compound in the biological sample, sir, actually, in the case of metals and minerals. Basically, sir, at the present level, sir, we are going to detect personally mercury, leads, or iron, anything. But in biological sample right now, sir, we are not able to detect mercury sulfide, yeah, iron oxide, and this is the basic issue, sir. When we are facing, when we are explaining our contribution to the scientific society, they ask about your mercury concentration is so much in the blood. But at the same time, we have to face, sir, we are using mercury sulfide, not we are using mercury at the our as our final product. Need your, sir, opinion and, sir, just guidance to, sir, how to proceed in all those things for identification of compounds, sir.
So I think, uh, one paper on Swarna Bhasma, where Swarna Bhasma was given and some blood chemistry was done to identify how much gold was available in the blood and few other organs for the body. I think for this what you are saying, Dr. Saab, your center, NIA Jaipur or even CCRS, need to conduct a proper study. Um, I will not say exactly pharmacokinetics, but something like that,
Yes,
where
sir.
you identify a patient, give any product to them, and then collect the sample, say after 7 days, after 2 weeks, after 3 weeks, and then develop the method which will be spectroscopic or ICP method, where you can test the particular compound whether it is present in the blood or not. I can only share you with Dr. Lavekar was DG. He started this project at Chandigarh, and to his surprise, he found that the normal population in Chandigarh was itself having very elevated level of mercury in the blood, which was totally unreported. Nobody knows the reason. But then that project was killed there only because if you are saying that we need to test the product, right? So sample zero should be zero in mercury. Then only you can give the product and test how much is gone to the body. But if the blood is already having mercury, then that subject is not good for you. So therefore, it needs I think Ministry of Finance to support a huge project on this, and with the NIA, with CCRS, you can work together, and you only need ICP-MS, which should be able to test the ICP-MS can test up to ppb level in the blood, right? So you identify what compounds you want to check, test one on baseline, and then test on, say, after 1 week, 2 week, 3 week. But then you need to admit the patient so that you can control what he is eating outside. Like suppose you do the study in Delhi, in All India Institute of Ayurveda, but the patient is staying in Sadra, right? So while going from that place to Sadhana, he will buy the green vegetables on Yamuna side, right? They are full of pesticides, they are full of heavy metals, right? So you will unnecessarily blame the Ayurvedic medicines while these are coming from the actually the vegetables what he is taking. So therefore, it's a very difficult but very valid research. To do it, you need to control everything. जो खाना भी खा रहा है, सर मैं बताता हूँ आपको, we are working on a project on phytopharmaceutical with Indian Council of Medical Research, and it is a sort of allopathic protocol for phytopharmaceutical. So when you are conducting the pharmacokinetic studies, one of the compound we were testing was available in the placebo group, which was not should have been in the blood. It was available in the placebo group. So our expert committee was just on the verge of rejecting our data, but then we thought of how can you reject our data? Have you checked the food what the patient is taking? Luckily, every patient was hospitalized. We checked all their food and we found their compound coming from the food. So therefore, if the compound coming from the food and the same thing you are giving your drug also, then also lot of this discrepancy will come and tomorrow people will say, my dear, you don't know how to do science. But that, my dear, you don't have right protocol. So therefore, also what you say very valid, and I think your institution and CCRS and All India Institute should join hands and do as I told you, Dr. Lavekar has once burned his hands, but he should not have stopped it. He should have continued some other city, right? So because what you know, first of all, also you need to do the survey of the normal population of at least 1,000 to see what is the blood level, normal basal level, and then you give the product. And also you look at the food, what food they are taking. So also when you do stratification, you need similar diet, similar basal level in the blood, and then you give that patient your product and then go on sampling. Otherwise, you can start from animals also. Because it is difficult to control humans, so you can start from animal. In your Rasashastra, Bhaishajya, Kalpana, you can tie up with some department and use it on animal, because in animal it is much easier, you can control the food also, everything is controlled. So give to the animal and then check in the blood. Make it the part of the toxicity study. So tox on one side, organ on one side, blood on the other side. Unfortunately, whatever toxic studies has been done, you might have seen CCRS has published 5 volumes recently on Rasashastra safety studies. Right? If you look at those studies, this particular part is missing there. But at least they have done a good job that they have published the safety studies on some, I think 50, I think some 20-25 products they have published. 5 वॉल्यूम जो आपने चेक कर लेना, अभी CCRS में पब्लिश किया हुआ है.
Thank you, sir.
Right.
Thank you. In between our Dr. Subhash sir also connected. Sir, आप कुछ बात करना चाहते हैं, डिस्कस करना चाहते हैं, सुभाष सर? I think he is disconnected. Sir, there is a request for your PPT also, sir. If you can please share.
Hello, Dr. Karuna.
जी सर.
Dr. Karuna.
हाँ जी सर.
May I ask something? Thank you, sir. Here I am, Dr. Sanjay.
How are you, sir? So nice, sir.
Thank you, sir. Thank you. Sir, as you told regarding the question of Dr. Rakesh, if I try to do the ICP, ICP essay only detect the level of mercury.
No, sir. It is any metal you want.
Okay.
Any metal you want. You decide what you want to test.
Suppose, sir, we are giving Rasindu to the patients.
बिल्कुल.
And that take the blood samples and go for the ICP. So ICP detect only the level of mercury, not the mercury sulfide.
No, mercury separately, sulfur separately.
Okay. Then, sir, we are always telling that we are not giving as a elemental mercury or the sulfur. We are giving as a whole compound.
No. What we are saying is that when you are giving mercury as sulfide, it does not get absorbed in the body.
Yeah.
So while you will be getting therapeutic benefit, you will not find mercury in the blood because it does not get absorbed. That is why it is safe.
Yeah.
It will go. So if you test stool also, actually, you should test the stool of the patient also. You will find it will go through via the stool or urine. There is no mercury you will find in the blood.
Yeah.
So that's why we should not look only blood. We should look other excreta as well.
Yeah.
Urine and this also.
Right, sir. Okay. Thank you, sir. I will try. I will allow some such type of topic to our scholars.
Sure, sir. Because see, if you don't do it, then who will do it? And this is very sensitive. As I told you, a very difficult topic because Dr. Lavekar, CCRS are working on it. and they found that most of the people in Chandigarh were already very high on mercury, so he dropped the project. But I think you should continue working on it, and in order to control, you can start from animals first, because there you can control the diet also, you can control everything, and look at the excreta also. Normally, if people have done also, they have looked only at the blood, they have missed the excreta. Because unless you prove, if though it is not in the blood, it is going to excreta, then your contention is very good. That though I am giving mercury, it is not going to the body, it is showing its effect and just thrown out through the excreta, through the stool or urine, and show how much you have given, what was the content, total mercury you gave to the body, and what is the content of mercury which went out through the excreta, and calculate that. Then only it will make sense.
Okay. Okay, sir. Thank you for your expert opinions. And
Thank you, Doctor.
we will take your advice, sir.
I am always available for any kind of help or guidance if need be.
Okay. Thank you, sir. Thank you. Welcome. Over to Dr. Karuna.
Thank you, sir. Sir, there is one more question into the chat box. Actually, the session is very interesting and people are getting. He's asking what is the maximum level of preservative percentage can be used in Ayurvedic products, especially in the decoction?
0.22%.
Okay.
Parabens 0.22% maximum.
Thank you, sir. Anyone, anything else? So Dr. Subhash Yadav sir from our department has requested for your PPT. If you can please share.
PPT, I'll send Dr. Anupam.
Okay, thank you, sir. Thank you so much, sir. Now I will request Thakur Rakesh Singh sir for vote of thanks.
Thank you, Dr. Karuna. On behalf of the National Institute of Ayurveda and Department of Rasas, myself, Dr. Rakesh Singh, Assistant Professor, Department of Rasas, Research Kalpana, I'm giving an opportunity to not only thank, to do my respects to our Dr. C.K. Katiyar sir, a name with a person and science from the Rasas and pharmacology. It is very rare actually. So definitely, sir, with your blessings and with the supports of the NIA and Department of Rasas of Research Kalpana, we are organizing these events, and definitely it's looking very fruitful as the suggestions and discussion coming apart from our student sides, from our definitely faculty sides. Definitely it was very, always it was interesting to see you and speak and listening your points regarding the different contamination of the raw materials. As I got your guidance from since I was in BHU, I think that you are always concentration on the raw material microbial loads. There are 5,000 microbial loads there. Still, you are going to check your final product. You will get more than 5,000 microbial loads. But still, that medicine is working in the patient. It's a very interesting and definitely, sir, your expert guidance. So, sir, once again, thank you for sparing our time. And definitely, you are enlighten us regarding the same suggested topics. With due respect, I also convey my vote of thanks to our Vice Chancellor, sir, Pro Vice Chancellor, and our Department of Head, Professor Anupam, sir, to organizing these events and connecting this different department of Agad Tantra and Department of Dravyaguna with us to just encouraging us to regarding to just substitute what is the basically lacuna with us and where we are and where we need to go. Actually, this is the first slide actually which I saw in your slides. So definitely, it is a very important point. And definitely, and it is a very learning point for all of us because the Department of Rasas is definitely we have to do work on the same lesson and plans. At the same side, I convey my thanks to our most respected senior faculties of our department, Dr. Rajendra Prasad Sharma ji, Dr. Sanjay Kumar sir, Dr. Moharpal Meena ji, Dr. Sakita ji, my colleagues, Dr. Ritesh ji, Dr. Subhash ji, Shobna ji, all my dear students from MD, PhD scholars from all the three departments, because your auspicious presence always enlighten us and give us chance to explore ourselves with the help of our expertise. Definitely, sir, in my thought, in my opinion, soon we will again hear you. It's my regards because always it was here too well. Sure. Please enlighten us, guide us your path. Once again, sir, from my side, from my should, thank you for giving your spare time with us. Thank you so much, sir.
Thank you. I am privileged to be with you.
Thank you so much, sir. Thank you. Namaskar, sir.
Thank you so much, sir. बहुत बहुत शुक्रिया. I also extend the thank you to all the participants, all the IT support and departmental support staff. Thanks to one and all. बहुत बहुत शुक्रिया, sir. We'll see you all on next Wednesday. Thank you.